Hello, my name is Claire Rosenbaum. I'm a third-year veterinary student at the University of Minnesota. Today I'm presenting on the identification of a novel papillomavirus in a mallard duck with mesenchymal dermal tumors with cartilaginous differentiation. Papillomaviruses are largely epithelial trophic and are known to cause papillomas, fibropapillomas, and carcinomas. Multiple papillomaviruses have been identified in avian species. This is a case report of an adult female mallard duck that presented to a wildlife rehabilitation center due to inability to fly. She was euthanized due to two sizable, firm, well-demarcated masses, one being on the second digit of the right foot encompassing the distal phalangeal joint, and the other being in the subcutis and musculature of the axillary region of the thorax. The veterinarian at the wildlife center removed these masses and fixed them in formalin. She then submitted them to the University of Minnesota's Veterinary Diagnostic Laboratory, where we performed histological examination, transmission electron microscopy, and next-generation sequencing using the Illumina platform. This slide shows histomicrographs from the foot. As you can see in the lower magnification image on the left, these masses showed multilobular organization, with the lobules containing well-differentiated chondrocytes, and the tissue surrounding the lobules showed aplastic spindle cell populations. The mass on the thorax had very similar organization with an increased aplastic characteristics. You can also see that though the distal phalangeal joint is encompassed by the mass, it is not infiltrated. Similarly, there's multifocal infiltration on the epithelial cells. However, there is no hypertrophy or hyperplasia of the epithelial tissues. On the higher magnification histomicrograph, you can see there are basophilic intranuclear inclusions within the well-differentiated chondrocytes. This was our initial suspicion for viral involvement in this tumor and was what caused us to further pursue transmission electron microscopy. This electron micrograph shows the viral particles within the chondrocytes. They had an average diameter of 44 nanometers and had a paracrystalline array. Based on the size of these viral particles, polyomaviruses and papillomaviruses were our top two differentials. Also, the presence of these viral particles within the nucleus of the well-differentiated chondrocytes suggests active viral replication within these cells. To further differentiate which of the two viruses was present in this mass, we performed next-generation sequencing. The sequencing revealed that this was, in fact, a papillomavirus. It contains the E1, E2, L1, and L2 coding regions, all characteristics of papillomaviruses. It also contains the well-established E6 coding region, as well as two hypothetical protein coding regions, whose function is not yet established. Overall, this virus is very divergent from all other established papillomaviruses. It's most similar to that of the Fulminars glacialis papillomavirus 1, with a 59% similarity in the L1 coding region, which by ICTV standards would define this as a novel species of papillomavirus. While certain papillomaviruses, such as the bovine papillomavirus, have caused mesenchymal proliferation, it's always with epithelial proliferation. This is only the second case report of a papillomavirus causing mesenchymal proliferation purely with no epithelial proliferation seen. The other case report was in a northern fulmar that presented with the Fulminars glacialis papillomavirus 1 previously mentioned as the closest relative to this papillomavirus. This case report raises the potential that there could be more papillomaviruses with purely mesenchymal proliferation. This means that there's potential for redefining the tropism of papillomaviruses to include purely mesenchymal differentiation. Further investigation into the hypothetical protein coding regions is also needed to identify what protein they would be associated with and if that protein may play a role in the tropism of this papillomavirus. Overall, this case report raises questions on the tropism of papillomaviruses and the potential that mesenchymal tumors could in fact be associated with other novel papillomaviruses. Thank you.